Hello, my name is Rahul Pandit. This is a video abstract on our paper, Instability of Spiral and Scroll Waves in the Presence of a Gradient in the Fibroblast Density, the Effects of Fibroblast-Myocyte Coupling, by Zimik and myself. A mammalian heart is an efficient electromechanical pump that supplies blood to the rest of the body. I show you a virtual human ventricle here, which we use in anatomically realistic simulations of electrical waves in the human heart. The contractile activity of the heart is mediated by electrical waves, which are generated by the sinoatrial node. When we have such waves, then we have the possibility of arrhythmias, which are spatiotemporal organizations in the forms of spirals or scrolls, and these are associated with life-threatening cardiac arrhythmias like ventricular tachycardia and ventricular fibrillation, which are leading causes of death in the world. I show you here a single rotating spiral wave. This is associated with ventricular tachycardia, and here is a broken turbulent spiral wave which is associated with ventricular fibrillation. Some studies indicate that episodes of ventricular fibrillation are preceded by ventricular tachycardia. It is important to understand this transition, and we do so by studying it in state-of-the-art mathematical models for cardiac tissue, which include myocytes, which are excitable cells, and fibroblasts, which are passive cells. In particular, in this study, we examine how these waves are associated, or rather broken up, by gradients in the fibroblast density. First, look at the case with just individual cells. In the absence of any fibroblast-myocyte coupling, we get an action potential of the sort shown in red, which is how the cell responds to a stimulus, and when these fibroblasts are added with different resting membrane potentials, this action potential is modified. In particular, its duration, the action potential duration, changes. We carry out simulations in 2 dimensions and 3 dimensions with various different distributions of fibroblasts. Let me just concentrate on one here, where the density of fibroblasts changes linearly as we go from here to there. Here it is high, here it is low. Roughly speaking, what happens is that the local frequency of rotation of the spiral wave depends inversely on the action potential duration, and then the wave, as it tries to travel in this medium, breaks in the region where this local omega is low. I show you one such breakup in a 2-dimensional simulation. You see initially nothing is happening, but then finally the wave breaks in the low omega region. And this is not a mere artifact of 2 dimensions. It occurs also in 3 dimensions. I show you a simulation from 3 dimensions, and you will see that this one breaks because there are more fibroblasts to which the myocytes are coupled. You see that here the wave is breaking again in the low omega region of the simulation. Similar breakup occurs if we pace the simulation domain at one end. Again, with enough pacing, the waves break in the low omega region of the simulation domain. In fact, we can draw a phase diagram which has pacing cycle length on the vertical axis and the resting membrane potential on the horizontal axis. In some regions we get no waves, and in some regions we get this re-entry, that is spiral waves. In conclusion then, we can study with great precision the effect of the gradient in the fibroblast density on spiral waves and scroll waves in mathematical models for cardiac tissue, and we hope that our study will lead to experiments on such systems, analogues of which have been carried out by varying ion channel densities, but never by varying the fibroblast densities. I thank you for your attention.